question. Do I need a microphone, or is this, I'm talking mostly to you? I do need a microphone?
No. Okay.
Huh?
No.
Good? Amazing. Okay, so I was told to speak about the CRISPR revolution, but another title would be, what was it?
Genetic engineering.
Genetic engineering, past, present, and future, could be summed up as CRISPR, the revolution, so that's okay. A quick background about me. My name is Gita Reinitz. I have my master's degree in biology, biotechnology, genetic engineering from Bar-Ilan University. I'm very passionate about going out and teaching people who don't necessarily have biology backgrounds what's going on today in the field, the things that sound exciting, why they might be less exciting, the things that sound scary, why they're not scary. Hint, it's related to why the exciting things are less exciting. I'm very passionate about education, and that's why I joined Ilya and Keren and a lot of great people in the BioHub IL project. I'm also a recent entrepreneur. I bring this CRISPR technology, the Cas9 protein, the RNA that we're going to talk about today, to Israel, and I supply it to labs at hospitals, universities, and companies while offering support of how to plan their project. BioHub IL, we definitely discussed. This was really fun. We have a space in Yigal Allon in Tel Aviv at a place called The Cluster, and I gave a course there. It was a 10-lesson course, 2.5 hours on a Tuesday evening, and I was shocked to find that people actually showed up to hear in-depth stuff about biology. And I said, this isn't enough. We got to get it hands-on. When I say me, I mean Keren and Ilya and 1 billion people, because takes a lot to get together. As you can see from the fact that they're trying to fight with their pipettes, these people have zero prior experience. Anybody can come. But by the end of it, I think that they really learned a lot. And so these meetups are a great way to get people interested in science more, to get these tastes. Before we talk about genetics, past, present, and future, we have to understand a little bit about biology. I understand that for the most part, I'm not speaking here to a crowd of veteran biologists, right? Let's see how we're doing here on time, by the way. Yeah, we're good. So I wanted to just give a rundown of some terminology that we're going to be using, of some facts that you may or may not have known. There are 37 trillion cells in the human body, give or take. In every single cell in our body, you've got a nucleus in the middle. Everybody's thinking back now to their elementary schools, high schools, right? Think about it. You've got cells in your eyes and your tongue. You have your nerve cells, heart, liver. Each one is identical in the sense that they each have one nucleus in the center. They each have, in that nucleus, 2 copies of DNA. Everyone here got 1 copy from their biological mother, 1 copy from their biological father. And what this DNA actually is just a bunch of, we call them letters, right? But they're really these molecules that encode for almost everything that we are. It's kind of like our source code, our operating system. And in that operating system, you have apps. So each app would be a gene. That's the functional pieces of DNA that tell you, are you going to be short or tall? Are you going to have blue eyes or brown eyes? What risk do you have for certain diseases? If you were a plant, how big and what kind of fruit are you going to be producing, if any? What kind of, do you need carbon dioxide? Do you need nitrogen to live? Your DNA, your source code is going to determine what characteristics are going to define you, obviously to a certain extent, right? Humanity. Cool. The genome is a very fancy word for all the DNA of an organism. So you have the human genome, a bacterial genome, a plant genome. What that means is the functional DNA, the apps, the background DNA, the regulatory DNA, the stuff that all works together. When you put that all together, all the genes and all the upstream, that's called the genome, right? The human genome, you might have, I don't know who in this room could remember, I personally cannot, but the Human Genome Project, right? That was completed in 2000, but it was started in 1990. It took 10 years and billions of dollars to sequence the whole genome of 1 person, okay? Today, you can do that in a couple of weeks for a few $1,000s, and it's dropping all the time. But what we're actually talking about is just all these letters that make up who we are. And what I found really weird when I was learning this is that 99.5% of that genome is identical between every human on Earth, right? So people who were born in China, people who were born in Africa, people who were born in Israel, 99.5% of every single letter, where it is, what it is, it's the same, okay? The things that make us different, the things that make us, you know, tall, short, this kind of skin color, this kind of risk for this disease, is 0.5%. That's the difference between us. Math is not my thing. Biology is my thing. In case anybody was wondering. Okay. Now, everyone here has heard of the term mutation, right? Genetic mutations. I think at this stage in the game, it's definitely entered into common. Wait a minute. Hold on a second. Is there a problem with the Facebook Live? Is that what we're discussing? Yeah. Okay. I have very severe ADD, and this chatter is very bad. But the Facebook Live is important. Is it working now? Are we live? Thank God.
I'm live.
Yeah. Okay.
It's okay.
Everyone, don't worry.
Yeah.
Your purpose in this room is important, but not as important. So Facebook Live, I had to check. Thanks for waiting. Okay. So genetic mutation is also a fancy word for when the code has been changed, when the DNA code has been changed in some way. It usually, that's the denotation. The connotation, what we use it for, is to mean something bad. Some mutations are great. They help us survive better. When we say genetic mutation, we generally mean the code has been changed and it's not a good thing. Something was mutated and makes a person more prone to certain types of cancer. There was a mutation at birth and now there's this type of a disease. It's usually not a good thing. Being a redhead is a mutation. I think it's great. But it means it's very bad for me to be out in the sun too much. It's a mutation that occurred a long time ago, back in Scotland, England, places that didn't have that much sun, so redheads weren't at risk of being burned. And what we got as an advantage was we could process sunlight much better than anyone else. So we needed much less sunlight to get a lot of vitamin D. It was a great mutation until you make aliyah, and then you hate it, right? So I don't go out very much. So genetic mutation is when the code is changed. It can be inherited, meaning from birth. It can be spontaneous, meaning we haven't figured out why it happened yet, so we say it happened for no reason. It can be environmental, age, lifestyle, sun. The sun can cause mutation, X-ray. Does anyone find it odd that I put age under the format of environmental? Or is everyone cool with that? Okay. When I say environmental, I mean something that you weren't born with. Okay? You can be born with an inherited mutation from your mother or your father, or it can occur over time from the environment. A cell's environment changes with age. Think about the DNA always has to copy itself. You have to get new cells all the time. Eventually it gets tired. You start to make mistakes with each copy, with each level. It was my birthday last week, so I'm getting very philosophical about where this is all going. So some of these mutations don't really do very much. This letter was switched and it doesn't matter. Maybe it wasn't in a coding part of the gene. Maybe it got switched for something similar. Some of them really do matter and they can lead directly to diseases. And a lot of what leads to what is not really known just yet. We're still researching a lot of it. This is the DNA I was talking about. We got the classic double helix. And those are the apps, right? Each app is a gene that codes for something that does a function, like a protein. And these genes are in a long string of DNA called a chromosome. We have 23 pairs of chromosomes in each nucleus, in each of our 72 or 37, I don't remember, trillion cells in our body. 2 copies, one from the mother, one from the father. If you were born a female, then your last pair would be 2 X chromosomes. If you're a male, you'll have 1 X and 1 Y. Other than that, the other 22 are pretty much the same between the genders. And every single living thing, from bacteria to plants to us, has this DNA, okay? This chair and the rocks outside, no DNA. A thing that can self-replicate and reproduce, yes, DNA. That's how you would know. And like I said, they code for your height and your eye color and all kinds of things, 1 copy from each parent. Then you transmit half of your DNA down to your children. Sometimes during that process of transmission, a mistake might occur. That would be a type of inherited mutation. Okay, so I hope you all were drawn back to your high school. I'm really great on time. This has never happened to me. I'm going to stop checking time altogether and see how that works. So now the question really is, what's CRISPR? By a show of hands, who in this room has heard of CRISPR? Okay. By a show of hands, who in this room has studied biology past a high school level? Okay. By a show of hands, who in this room works or has worked in the field of biology within the past 2 years? Good. Those are hands that are making me feel better. Because what I want to actually see is the fact that the majority of this room does not come in day-to-day contact with biology, yet the majority of this room has heard of CRISPR, right? Who here has heard of CRISPR from some sort of non-science source, popular media, a newspaper article, social media? When I say the thing that made you hear about CRISPR, you can raise your hand and keep it up, okay? So, social media, okay? A newspaper, not a journal like Science or Nature, okay? A television show, YouTube, Friends. Where have you guys all heard about CRISPR from? I'm not like hitting on it. Scientific journals? Has it been? Are you guys reading Nature? Okay, we'll talk. University. University. Who learns about CRISPR at university? Yeah, barely. CRISPR's only come out a few years ago. Okay, this is very nice. We'll discuss at the end where this is all from. But the question is, everyone has heard of CRISPR for some reason in this room, but what is it? Okay? The truth may shock you. Click on this. So, we all know that we have in our bodies 2 different kinds of immune systems. We have the kind that we were born with. The reason that babies can survive their first few days and weeks is because they have some sort of rudimentary immune system. But then you also have the kind that you pick up over the course of your lifetime. The easiest example is a vaccine, right? A vaccine is taking some sort of a disease, but a weakened version of it. It's putting it in your body, and it's weak enough that it won't kill you, but it will activate your immune system. Your T cells, your B cells will see it and say, what is that? I'm going to store this for later because this doesn't look good to me at all. And then if that virus comes back again in a real way, the strong version, your body can react very quickly because it's stored that information away, and it knows how to deal with that particular pathogen. What's very weird is that bacteria also have these 2 systems. Okay, bacteria, a one-celled little thing that makes you sick or makes your dog throw up on the carpet, they have an adaptive immune system. What we're looking at here is this blue thing is a type of it's a model, it's just a drawing, right, of a bacteria, and it's being attacked by a bacteriophage. That's a type of really tiny virus that attacks bacteria. And that bacteriophage, you see that diamond up there, it's inserting its own DNA into the bacteria. Here's the bacteria's own DNA, the genome, and this is the viral one. Chances are that bacteria is going to die from the bacteriophage. But if it manages to save itself, the way that it does it is like this. It's going to cut up the viral DNA, put a piece into its own genome, and keep it there. Now we've got this little red guy that it didn't used to have before. That's its memory. Just like our B cells and T cells take that vaccine and remember it for later, bacteria know how to do it too. And our B cells and T cells are always patrolling our body, patrolling our bloodstream, looking for the pathogens again. Same thing here. The source code is making the apps, and they're going to be patrolling. If the virus comes back again, there's no chance that this bacteria is going to die. The CRISPR-Cas9 system of the bacteria is going to recognize this DNA and cut it up into little bits. Okay? The system was developed and evolved purely in bacteria, purely to protect itself from future viral attacks. Okay? It's its adaptive immune system. It wasn't born with this knowledge. Because a virus touched it, now it knows how to protect itself in the future. That's the big deal about CRISPR. Thank you so much for your time. And no, what's cool about it is the fact that we took that knowledge and we used it for our own purposes, right? We said, what this actually is a type of protein that cuts DNA in a very specific manner. Maybe we can take this and make cool tools out of it. Maybe we can make medicine. Maybe we can learn what genes do. This has become a tool. What we use CRISPR for, instead of an adaptive immune system for bacteria, which is really boring, frankly, I don't really want bacteria to survive most of the time. What I can use it for now is gene editing. I can use CRISPR to manipulate endogenous genes. Endogenous just means naturally inherited, the genes that I have in my body. okay? As opposed to transgenes where I took something and made a crazy monster. Okay, so how does that work? We said we have this genetic code, right? We call them T's, C's, G's, and A's. Adenosine, I don't even know if I remember these, honestly. And guanine, cytosine, thymidine, and adenosine. Okay. This is our source code. And then we take something like CRISPR and we give it instructions. It's almost like, you know, on Microsoft Word, you can do Control F and it's going to find what you're looking for. So you tell the CRISPR, listen, I want GCA, GCT. Okay, no problem, it's right here. My only input is telling it which letters I'd like to target. And then the Cas9 can come, that's a type of protein, and cut the target that I asked it to cut. Again, it was developed to cut viral DNA, but now it's cutting whatever DNA I tell it to. This might be DNA in the nucleus of a cell in a mosquito, in an apple, in a person, in a whatever you want. I'm actually really happy that Tali's here. Wasn't sure if you were coming. But I was looking for a long time on YouTube and on places for just some sort of visual, nice video that shows what CRISPR is doing in a simple way, not 7 minutes. Maybe today it exists, but 4 years ago it didn't. So I sat with Ori one weekend and cut up a bunch of things and made a stop motion. And then 3 years later I met Tali, an animator, who made it look much more normal. So this is a combined effort where I just want you to just visualize what it is that CRISPR is doing. Oh, what? You don't have your sound off like I do. And that's shameless advertising of my company, CRISPR. Okay. Beautiful. Okay. Here's the DNA that you guys want to cut. DNA has 2 strands. Cas9, right? You always hear CRISPR, Cas9, CRISPR, Cas9. Cas9 is a protein that knows how to cut DNA. That's the only thing it knows how to do. And then you've got this RNA that tells the Cas9 where are you going to cut. That's the search and find we were talking about. They need to come together, scan the DNA, and when they find a match, they're going to cut it. Watch how it happens on this animation. If I could find the mouse, you could watch it. Okay? The Cas9 says, where am I going to cut today? Let me know. They scan. Here's a part that looks the same, and boom, you have a double-stranded cut. And Tali is awesome. No, but really. So it started off as an adaptive immune system for bacteria, and now we know that CRISPR actually cuts DNA in a really specific manner. It's not random cutting like UV lights that just make it go super highly targeted. Still, I don't understand why this is the biggest revolution of the entire 21st century, right? There's a common misconception that I've been seeing, and actually a lot of articles, a lot of things written about CRISPR, that says CRISPR is the technology that erases genes, or CRISPR is the technology that replaces genes, or inserts genes. This is absolutely not true. CRISPR is a technology that cuts genes. That's all. It knows how to cut. The reason it's so amazing is because in our cells, there are all these different mechanisms that will react to those cuts. Okay? DNA damage is the worst thing that could possibly happen to a cell. You don't want a chromosome now flying unattached to itself. When you have that cut, a cell will respond and quickly try to repair itself. Now, what happens when you're trying to do things very, very, very quickly, generally?
Mistakes.
You make mistakes, right? Because haste makes waste. Is there a Hebrew version of this? You were helpful before, so, okay. Anyone? Yesh mashu?
חיפזון מן השטן.
Which is what? חיפזון is a snail.
לא, חיפזון זה...
Rush
is from the devil.
Rush is from the devil.
Satan is the devil.
rush is from the devil. That's much better than haste makes waste. Okay. So, rush is from the devil, and fixing your DNA quickly will probably lead to mistakes. That's why CRISPR is so cool. I made damage at a very particular spot with CRISPR. My cell fixes it, but it forgets to put in one letter. I've now shifted the entire code. Is anybody here a coder or a programmer? Or have you ever learned a little bit? Who here has wasted over 10 minutes due to like a comma or, okay, so that's this. You put it all together, you lost one letter. I don't know how they taught biology before computers were a thing, because it's really, the resemblance is striking. And that's what's so cool about CRISPR, right? It's kind of that catalyst of saying, I'll put the damage right here, i.e. I'll put a deletion right there. Because when the cell fixes it, that's what's gonna happen. Most of what's cool about CRISPR is actually the way that the cell reacts to the damage. It's not really the CRISPR itself, though we need it, right? This is what the videos used to look like, because, you know, I didn't want to kill Tali too much. So this is the stop motion that we actually made, where I wanted to show you what this mechanism kind of looks like. This is the DNA that's just been cut by our CRISPR, if you remember. And the mechanism is called non-homologous end joining, NHEJ, where proteins that are already in the cell, you know, we didn't do anything fancy. That's an exonuclease. They come to this damaged piece and they're gonna start to chew away at either end. Stop motion, very primitive, okay? Until there's a little bit of homology, they match up and they can come together. A ligase is an enzyme that's like glue, it'll put it all together, and that is what's so crazy about CRISPR. The CRISPR cut it, the cell fixed it, and look, it's missing two shapes, right? Okay, that's the difference between a functional protein in a gene and a completely not functional one. That's the difference between, well, I'll give you a lot of examples soon. Okay. I'm not going to go into this one too much, but when you hear that CRISPR can insert a gene or replace a gene, what they're talking about is this: a different cellular mechanism. One mechanism says non-homologous end joining. These ends don't really match. I'm going to chew them away and put them together until they do. I'll do something just so that the DNA is not broken. That causes a deletion a lot of the time. Homologous directed repair is another mechanism in the cell where it's a little bit more elegant. You know what? I'm gonna show it to you. Okay? What happens here? Can I just? No. Can't just press play like that. Jenny, thank you. Okay. So what happens here with homologous end joining? Here's the CRISPR. I simplified it because I got tired. Here now you have this cut, and instead of the cell replacing, fixing it quickly with deletions, I'm adding a new component to the cell. Okay? But this is work that I have to do. I'm putting into the cell the CRISPR to cut, and I'm also going to put in some sort of a replacement. Again, remember, the cell just has this broken DNA and it wants to fix itself. If it finds this really nice template that matches exactly, you see how nicely it matches up, it's going to say, oh, this is probably the other copy of DNA. I know this guy, he's cool. I'm going to swap it out. I'm going to fix myself using this as a template. Okay? Instead of chewing away and sticking it together, they're going to use this template, which sneakily I put in there, and they're going to swap it out with this. They think that this is a fixed version of themselves. What is it really, though? What did I put here in the middle? I actually snuck in a gene that I wanted to be in there. That gene might just be a correction of some mutation. By accident, there was an A in the middle. I want it to be a T. I'll put in this whole thing that's exactly the same, but here there's a T instead of an A. The cell will replace that part of the DNA, and I've now fixed a mutation, okay? So keep in mind, the only thing CRISPR did was got us started. Now, I'm sure you're all very frustrated at this point because I'm telling you a lot of really interesting facts about CRISPR, but not a single one of them is justifying the buzz that it's getting, right? You're like, oh, yeah, I switched to T, right? And you're getting very impatient, but I still have time here, so I'm getting there. Why is CRISPR such a big deal? It was actually discovered in the 1980s originally. They said, there's a weird pattern in the DNA of this bacteria. This DNA is in a bacterial genome, but it looks like viral DNA. And not only that, it keeps repeating itself. It looks almost like clustered, regularly interspaced, palindromic repeats, like a CRISPR. And next to it, there's this gene that cuts stuff, and we're going to call it Cas9 for very complicated reasons. But we have no idea what it does or why it's useful. It's a thing. They discovered it in the 1980s. Didn't know it was an immune system, didn't know about the cutting and this. It was only in 2012, which was basically yesterday in terms of biology, in terms of most fields of science, right? It was only 2012 that they realized what this tool could do, and they started to use it in new organisms. They said, why should bacteria have all the fun? Let's isolate this protein and see if it can work in a human cell, and see if it can work in, right? This only happened in 2012, so it's very new. That's one reason it's exciting. But you also need to keep in mind that if anybody wanted to replace a letter in the DNA before 2012, they would either rely on random mutagenesis, which basically is like trying to, I don't know, fix your wall by like throwing a bunch of random stuff at it and seeing what happens, right? So a bunch of x-rays and things that cause mutations and hoping the mutation you're looking for would happen. You can all agree that that's really not efficient. Then they tried something called zinc fingers, which also knows how to cut DNA in a targeted way. That's been around since the late 80s, exactly like CRISPR. The difference is that instead of the RNA showing you where to cut, it was a protein. The difference between RNA and protein is the difference between an electric bicycle and a Mercedes-Benz. Okay? They'll both get you there if it's close. But one is overkill and not everyone can really afford it, and one every 14-year-old in Israel is hopping around, right? So the zinc finger was the Mercedes-Benz. It does exactly what you need in terms of what CRISPR can do, but a very limited number of labs could use it. Also, making it would take months just to make that thing, which CRISPR can take a few days. Then they came out with TALENs, which is a heartbreaking story, because TALENs also does what CRISPR does. It's 1/10 the price of zinc fingers, okay? 90% cheaper, and it only came out in 2010. So it had 2 years in the sun of revolution until CRISPR just killed it, okay? Just absolutely destroyed it. Zinc fingers had a good 20-year run, 20-year run. Talens had 2 years. Extremely unfair, doubly. Okay, what we have with CRISPR, and the reason it's so revolutionary is because you can switch these letters, hooray, but you can do it quickly. Okay, this whole RNA thing, you find what you're looking for, you can order it online, and it'll come in from California or wherever you want. Within 2 weeks, it's here. It's modular. So with zinc fingers and talons, the thingy that cut and the thingy that guided was the one piece. So you had to buy that whole huge piece each time. Here, Cas9 protein, you just buy as much as you want. And then you just order this little RNA each time. It's modular, which also makes it cheap. The fancy word is accessible. But what accessible means is cheap, right? Everyone can access cheap things. Here's a really nice quote that I found. In the past, it was a student's entire PhD thesis, that's theoretically 4 years, but in practice much more, to change one gene, said some guy from San Francisco. And CRISPR just knocked that out of the park, okay? A first-degree student with a couple of months of time can edit thousands of genes, okay? And each gene probably costs a few $100s, which again, in your basement might be a lot of money, but think about it in terms of research, it's unbelievable. Actually, it could be even as cheap as $40, just less effective. Okay. Everything I just described is very cool, but so far you have to take my word for it, because honestly, there's nothing really that great going on. Revolution in basic research. Basic research is loving life. Why would that be cool for, The Economist and Time Magazine? Why is everyone talking about CRISPR? Inside our academic labs, it's called, I think, the Ivory Tower, everyone's celebrating. But what about CRISPR is making the whole world celebrate? Is making people like, who's that guy who invented Napster? Sean,
Penning.
Huh?
Penning.
Yeah,
Penning.
he's investing in this stuff. All these tech people are getting so excited and they want to know more about CRISPR and they want to find better versions of CRISPR. What's the deal? And when I say deal, I mean practical applications. Applications that can save lives, applications that can be very lucrative.
They have not studied biology.
Number one, it's don't study biology.
No, they have not studied biology.
Oh, okay, yeah. Well,
They
same,
don't
right?
understand how much of the genome we still don't know.
Right, and how non-lucrative that knowledge anyways is for 99.9999, right? Okay, for sure. But really, the question remains, we've been through, where did CRISPR start from? How did we get to where we are today? Why is every researcher so super excited? But why should you guys be excited? Why is this meetup so full that there are 3 people standing in the back because they don't feel comfortable sitting next to strangers because I actually see 2 empty chairs. But the point stands that while you stand, the point stands because it's an interesting, it's compelling, right? People are tuning in to what CRISPR is. And I think what most people came here to learn, and the reason I deleted half of this slideshow, because I put in so much stuff that I love as like a really technical, but I deleted most of it because I think meaning it, and I think that's what people actually want to know, not these diagrams of like DNA. So, why is popular media interested? So I'm going to give you some really cool applications. At the end of this lecture, my phone number is on the last slide, and you can just WhatsApp your email to that phone number and you can get these slides. So if these things interest you and you don't remember all of them and you haven't brought your notebook or whatever, you can get these slides and you can whatever topic interests you. I brought Is it millions with an S? I think it's million with a million, but that's a lot of people. Meaning, you guys have Shark Week in America, where sharks kill something like 40 or 50 people a year, maybe less. And no one ever goes for Mosquito Week, even though it's the single largest killer in terms of a living organism on the planet. It kills 1 million people every year. Is it more? Anyone? Google? It's rude for me to Google, but you guys are all on your phones anyway.
Light is better, right? We came too dark, right?
Okay.
Yeah, really.
1 million gratitude points for the person who tells me how many people die a year.
700,000.
Aw. That was 1 million. Is it still the animal that kills the most people, though? How much of this is a lie? Nobody even knows, because you trust me, because I have this mouse that moves the slide. But I just wrote these slides. Anyone can do this. So anyways, the fact remains, mosquitoes are very, very dangerous, okay? And the reason that they are is not the mosquitoes themselves, but the diseases that they can carry, the parasites that will infect the mosquitoes that will then infect people. So one such parasite causes malaria, which the mosquitoes don't mind one bit, but is very bad for humans. Researchers used CRISPR to cut a specific piece of mosquito DNA and to then insert 2 new genes that will make this mosquito resistant to the parasite. Now, did the CRISPR insert those genes? No, it did not, right? The researchers did, but the CRISPR is the reason that the genes could go in, right? They made that cut. The CRISPR made that cut. So this is very compelling. You basically take a few mosquitoes in your lab, you inject a protein and an RNA that tells it where to cut, you inject some new DNA, boom, you have these mosquitoes that a malaria parasite comes over to them and they're like, I'm resistant. This doesn't talk to me in any way. What's more interesting is that you can release these mosquitoes into the wild, aka a cage with untreated mosquitoes, and they'll reproduce, and 99% of their offspring are also resistant to the parasite. This is why it's relevant, because you can't really go around collecting every mosquito in the whole world and putting CRISPR into it. If it can't get passed on naturally, you haven't done a whole lot, right? As amazing as CRISPR is, there are limitations that you need to face. So one of the things we're doing with CRISPR is removing malaria from mosquitoes, saving up to and including 700,000 lives a year. That was application 1, 700,000, check. Okay. Another thing you can do is make crazy goats and dogs. I don't know, I was researching, it was very early in the morning, and I saw that China, because it's obviously going to have to be China, is using CRISPR-Cas9 to delete genes that inhibit muscle and hair growth. This is one of the hardest parts of biology. You have to take a philosophy major to understand it. They are deleting the genes that inhibit muscle and hair growth, aka more hair growth and more muscle. When you delete the inhibitor, you get more, right? Okay. So what you're basically saying is these goats have more meat and more wool. So that's, you know, hypothetically a good thing if you're not a vegan. Even if you are vegan, it's a good thing because you need less, right? Less of these animals to supply the horrible people of the world. So this is good news for vegans as well.
Also, Israel is a big thing, featherless chicken.
Israelis are making featherless chickens? That's excellent. I will enjoy my Haredi cousins' weddings much more in the future. They don't fuck very thoroughly there. That's cool. They have also done CRISPR editing on beagles, but that is top secret and no one in China is willing to comment on why. Okay, but stay tuned. China is aggressively doing everything that it does, but one of the things it's doing is aggressively researching genetic engineering and cloning. Cloning is also super compelling. It means that you take, instead of one set of DNA from a mom and one from a dad, both sets of DNA are from the same organism. It's not cloning where it's actually another me, but it's cloning where 100% of the genetic material is from one parent, not two. Okay, that's really exciting because a lot of, let's say, lab experiments work with mice that are sterile, and then when you need more of those mice, you're in a bit of a pickle. But if you don't care that they can't reproduce traditionally, you can just clone them from that. That's really cool. So I think that China thinks that the combination of genetic engineering and cloning, which sort of go hand in hand, if you've genetically engineered something to be sterile, you better know how to clone it, is something that they're really pursuing a lot, and I think they see a future in it for the livestock and agricultural industries. We'll talk about that also in a second. I'm so sorry for this picture, but it's so accurate for what I'm trying to explain. Another application is giving pig organs to humans. Weird, right? Everybody here knows that a lot of, what is it? The piggyback heart, right? Like pig hearts are the valves. Anyways, pigs are disturbingly similar to humans genetically and also in the shape of their organs, and they're very, very similar, but not similar enough. So there's this guy in Boston named George Church. He's one of the craziest scientists that I've ever heard of and recently got to meet, so ever met, he's the guy, have you heard, who here has heard of the woolly mammoth project, that someone's trying to bring back the woolly mammoth to be alive right now? George Church, that's him. Okay, so in addition to making a real woolly mammoth, I don't know how he got the funding, this guy is saying, I'm going to use CRISPR. He's also one of the original discoverers of CRISPR, but much less discussed because he's not part of the lawsuit. He said, hey, pigs are super similar to humans. You know what? They should be identical, right? So he located 62 genes that he said, if I manage to edit these 62, certain tissues of a pig will be indistinguishable from a human. I'm going to make humanized pigs. Not only, I don't even know what I was saying anymore. I'm going to humanize pigs, okay, for the purpose of organ transplants. By the way, this couldn't be coming at a better time because self-driving cars are going to be causing a large shortage of organ donations. That's not a joke at all. That's not a joke at all. Most people die of old age or disease, not ideal organs. Motor vehicle accidents often involve healthy young people, and if you get to them quickly enough, and they wrote that thing on their license that allows for it, a lot of organs come from that, and it's sad, but it's also really, really important. And the more self-driving cars that are going to be on the road, which is happening, the less accidents there will be, because cars don't SMS. So I'm hoping that these technologies collide, and that we can simply, grow, maybe not in Israel, because there's issues here with pigs, right? But we grow these humanized pigs as this great source of our own organs. Again, vegans, if you don't agree with this, come on, people are better. Okay, so this is also a really big step forward because today, 99.9% of researchers are using CRISPR 1 or 2 or 3 genes at a time. It's much simpler that way. This 62 gene thing, it's extremely, what's the word? Ambitious. It's extremely ambitious, but so is George Church. He's about 7 meters tall. It's just his thing. Okay, so that's another application. Oh, I skipped 4. Okay, another application, probably one of the most talked about, is developing new kinds of drugs. So hard not to say opinions, but pharmaceutical giant Bayer, ask me my opinions later, and startup CRISPR Therapeutics. So since CRISPR got discovered, a million little startups are popping up. okay? And the strongest ones are the ones run by the people who discovered CRISPR in the first place, right? So you've got Editas from Broad, MIT, you've got Caribou Sciences from Berkeley, and CRISPR Therapeutics is one of those, I call them the top four. Like those first four that started in 2012 have such a massive head start on anyone else. If there are VC people in the crowd, like that's where the money's at. So CRISPR Therapeutics joined up with Bayer in a $300 million joint venture to just research a ton of diseases. Imagine that you've been researching pharmaceuticals for 45 years, and all of a sudden someone said, here's a magic wand. It makes everything faster and clearer. You wouldn't focus on one disease. You would just go crazy, right? So currently there's heart disease, blood disorders, blindness. They're not focusing on every disease. Their first targets are going to be diseases that have known mutations, right? There's some diseases that we don't know what causes it. Those known mutations are at the DNA level. Sometimes it's not the DNA at all. The DNA is fine, but the epigenetics don't even. Okay? It's at the DNA level, and it's at a single gene, right? That's what we call the low-hanging fruit. They're going to choose the diseases where their mutations are known, it's at the DNA level, it's one gene, and that resides in an accessible organ, right? CRISPR is great, but how's it going to work if it can't get to the spot it's supposed to work on? Okay? So a disease in the brain where you have to have the issue is more delivery, right? So working on, for example, blood disorders is one of those low-hanging fruit. Because the delivery is right. Okay, fine. This partnership represents the beginning of a significant and aggressive period of research. I don't know what that one means. That could lead to many cures. So this is very exciting. I think that I have an example later on, but okay. Another very exciting application for CRISPR is in agriculture. Plants are very tricky, but they sort of sustain us. So when the environment quickly changes and suddenly there's not enough land or not enough water, or there's a new type of pathogen or pest in the air, you can actually have a lot of crops decimated very quickly. I don't think we realize how vulnerable we really are, right? We've got our houses and our air conditioning, and we're all very proud of ourselves. But what year was it, that like volcanic ash? Like 2010 or something, where there are just no airplane flights because lava. It just snaps you back to reality that we live on a planet and the planet has rules, right? So we're trying to break more and more rules all the time. CRISPR's helping, actually quite a lot. And now we can start to really play with these genes in plants. We hear the term GMO very often, genetically modified organism. It's like the worst thing in the world, and if you ever eat it, you'll probably grow a third head after you grew your second head. And obviously that's not true, but the problem with GMOs, the true problem with GMOs, is that popular media got to them before scientists did, and also that once the scientists did, they don't speak very well. They're like, oh no, it's just a transgene. We spliced and everyone's like, this is horrifying. So I think what's really exciting about CRISPR is that it's part of a millennial generation, and we're getting to build the story before everyone else has to. There's very little difference between a GMO and a CRISPR, but please don't tell anyone that. And we can see that because there's already a CRISPR food on the market. They edited mushrooms so that they don't turn brown. Yeah. They deleted the gene, right? CRISPR cut, and then the cell fixed it imperfectly in mushrooms that caused it to turn brown too quickly. Maybe they turn brown like eventually. And because there's no gene from another organism, which is GMO and horrible, but actually just something within itself, it passed regulation, and now people can buy it. I had a conversation with someone in San Francisco, and I said, you know what makes me mad? It makes me mad when investment goes towards making mushrooms not brown, because I don't care. And she said something that I have to share with everyone all the time because it was very important. She said, I know someone who made a product where it was an apple that didn't turn brown too quickly. Not using CRISPR, using traditional crossbreeding and waiting a million years. But but they made it. And she asked that person this question, you know, why did you invest so much of your life when, you know, there are real problems? And he said, it's like Inception story, that every year, thousands and thousands and thousands of pounds or kilos of food is thrown away because humans are dumb. A brown apple or a brown avocado is not bad, but they think it is and they'll throw it out. And if you can prolong the shelf life by a few weeks, then you can actually be saving tons and tons of waste. That's why. And every time you get an apple on your table, you know that someone had to pick it and deliver it, and there's gas and there's this. Make that happen less. Don't put it in the trash. Okay. So don't get mad at the non-browning mushrooms. It was also probably a low-hanging fruit, let's be honest. But it's cool, and you can do more. You can do more, right? You can change the genes so that they're less sensitive to pathogens, so that they grow faster, so that they grow better. I do want to leave time for some questions. So making super plants is cool. I think this is probably of the most famous examples. I'm going to stop assuming that I know what people know and stop saying that. This is a really cool example of what CRISPR can do. Eliminating cancer. It's compelling. How many, just out of curiosity, how many people here have heard of the term CAR T cells? You don't count, Jacob? You CAR T. Okay. Good. Here we're getting to a bit more ambiguous. The study of CAR T cells that actually worked, they used CAR T cells to cure leukemia in a little girl, and they actually used TALEN technology, not CRISPR.
Cure or delete?
Cure.
Cure is in the meaning that they still don't know where the gene came from, or delete, they know where the gene came, and they deleted the gene of the...
Cure.
Okay.
Cure, and I'll tell you why. That's a good question. You're getting a bit philosophical, but I would say cure. An infant with this type of leukemia was cured from her leukemia. I don't think that it means they know where the leukemia came from. Cancer is not a disease. It's an amalgamation of hundreds of diseases that come about for hundreds of reasons, and it's a crazy, huge monster. But CAR T cells can fight that before we fully understand what's going on inside. So that's why it's cured. This cure was made with talons. CRISPR is still better because instead of this one fluke story, we can now work in a really high throughput manner to get to those cures. Because like I mentioned, talons and CRISPR are almost the same thing, just CRISPR is much more accessible. What are CAR T cells? What's the idea here? Cancer is not like the flu. It's not like something external has entered your body and is messing up your systems. Cancer is an internal thing, right? It's the body's own cells, its own DNA, suddenly saying, I'm not a part of this system. I'm going to grow as fast as I feel like. I'm going to take as much resources as I want. I'm going to spread out and start growing here. It's crazy, but it's your own cells doing it. And it shouldn't be happening. You have an immune system. You have these T cells. You have these B cells. They're constantly surveying. They should be noticing that these cells have gone rogue, and they should be killing them. And a lot of times they do. Apparently, I don't know how anyone could know this, but apparently people get cancer all the time, but only like a teeny bit, and for the most part, their immune system can take care of it. So cancer is not the problem. The problem is when it gets to stick, okay? Or in other words, when the immune system fails to recognize it. So what people are trying to do with gene editing technologies is trying to kind of smack the immune system in the face and say, recognize that, okay? How do we do this? You extract T cells from people's bodies, you put in CRISPR that will cut the DNA in a specific spot, and you insert a new piece of DNA. You put a new app into the T cells, okay? The app will code for a protein, a type of protein called an antigen. It sits on the outside of the cell and it kind of just surveys. Again, our bodies do this anyway. We have T cells anyway. They have antigens. They recognize a ton of great stuff. In this case, they didn't recognize the cancer. We're not very happy with them. So we're going to take them out of the body, cut it with CRISPR, put in this new piece so it can have this new antigen out, and now it will recognize the cancer. And it will beat it, right? So you put the cells back into the body. They start to go around looking for the cancer, they will now recognize the cell as a threat and they will kill it. Okay?
But only if
It's
there's
like an
any
information
difference
plugin.
on the cancer cells from the original cells. If you find some way that the cancer cells
Okay.
are different.
This technology works if we know which antigen is going to recognize it. And this is where a lot of the concept of personalized medicine comes in, right? Even within a subset of cancer, there are many subsets, right? So cancer, and then breast cancer, prostate cancer, blood cancer, brain. But even within those, there are thousands of varieties that we're only learning about now because we're able to finally sequence the DNA and see the differences and say, oh, oops, we thought the person had this just because it was in that physical spot in the body, how primitive. Actually, they have this, and this medicine will work the best. So we do need a lot more research for this stuff to work. The technology's here, and the technology's great, but we need to know which lock and which key is going to work for each type of cancer. I hope that was clear. Another big debate is economical, really. The cells I just described that you fix, are they going to be kind of an off-the-shelf type of cells, where you take one person's T cells, add this antigen, and then give it out to everyone? Or are you going to take my cells and put mine back in, right? And the pros and cons of that. I would rather use my cells. My body won't reject it, probably, because they're mostly the same. Most companies prefer I use off-the-shelf because, bulk. And they're playing around with both technologies now. Yeah, you guys can all see this, the current limitations of CRISPR are the fact that we don't know enough about genes, and that's really not CRISPR's fault. Also delivery, how do we get it into the places that we need to get it into? Again, not their fault. And the final thing is that I mentioned how once you damage the DNA, the cell will decide how to fix it. Will it do it imperfectly or with an insertion if you add DNA? But I wasn't fully honest on that. Just because I've added new DNA, it doesn't mean the cell's going to choose to do that. It takes a lot more energy. For every one time the cell will choose to insert a gene, there's another 999 that it won't choose to do that. And we don't understand why, and we need it to be a lot more efficient, right? All of this is basic research that needs to be done. I think that I'm going to wrap up in exactly 1 minute and have 5 minutes left for questions. But because I have you all captive, I thought that I would have the last slide be sort of a lesson or how I feel about CRISPR, because I really am obsessive about this technology. And when I think about it, here's what I see in my head. It's basically that these two women, Emmanuelle Charpentier and Jennifer Doudna, each with their own separate labs, were kind of just sitting in their labs doing very, very basic research. Jennifer's expertise is RNA, the structure. Emmanuelle works on microbiology, bacteria, and for 20 plus years they led extremely dull lives in their labs, just asking super basic questions. Neither of them were looking to discover a groundbreaking, crazy DNA technology that's going to save lives and make billions. They literally just said, what does this do and why? And if anyone here has done research, it's the most thankless work on the planet. And then one day they were at a conference, and Emmanuelle said, hey, Jennifer, I'm a bacteria kind of person. You're an RNA person. I found this weird thing, and they've been researching it forever, but I noticed something, I have a hunch about it. Can you confirm it for me, because your field is really RNA? Let's work together. Why did they do this? Because they were curious. That's it. Basic research. And thankfully, someone believed in their curiosity enough to say, oh, we'll give you money so you can look into this, right? You can have students and you can have giant lab equipment. Go for it. They didn't need to make like a business plan of what's going to be in. They just had their basic research. A lot of funding for basic research is being cut, and people want to understand the application, and people want your research to be really, really low risk. Like basically you have to guarantee it's going to work, which is not what research means. That would be more like manufacturing, and even that's not. And people need to realize that these bajillion-dollar technologies that everyone's so excited about, literally only exist because of basic research. And obviously the last lesson I get, which is like probably less relevant to most members in this room, is no, but that's sort of the point, right? Is the concept of empowerment and speaking to young girls about what kind of things they can do. And yes, we're in 2017 and everyone's very equal and it's great, and I'm very grateful for the opportunities that I've had. But when people start to laugh and be like, oh, why are women so up in the bed? The fact is, if we don't support the women of our society to be all that they can be, whatever that means to them, then we're all losing out. Because if these guys were told, you really shouldn't talk like that because you'll never get a husband, then nobody would have CRISPR, right? And I'm saying this is serious. It's not the type of feminism where it's like, oh, who really cares? This is practical, right? And if all of the women in Saudi Arabia can't drive any cars, maybe the cure for something is sitting in one of their heads. That should bother us actively, right? So those are kind of the lessons that I've learned. Really, we need to be encouraging young girls and young women as much as possible, and really we need to be supporting basic research, and these are not fluffy concepts. I've shown you really hard applications for why this is a worthwhile investment. It's just slow and people have no patience. That's basically where we're at. If you guys want the slideshow, I think that's my correct number. And two things are going to happen now. I'm curious as to which order. Okay, questions are going to come soon. The real final thing is that the reason we're here at a meetup and not sitting at home watching this on YouTube or on Facebook Live, which is where we should all really be because we can wear comfortable clothes, is because we're people and we're all going to be in the same room. So if everyone, the second the speech ends, just gets up and runs out, I truly feel you've wasted your time and your gas money or whatever else it is. A meetup is about getting to know one another, meeting one another. We already have something in common, right? Tuesday evening, we're going to learn about super random abstract things. I wonder what brought everyone here, right? That's a lesson I learned at BioHub throughout the course that I gave over. And particularly, just to guide you in that networking that I really would love it if everyone kind of just did, just to guide you, is there are a couple of those class members here, I won't tell you exactly who, and one of them, who I had the pleasure of meeting, called me after the course and said, Gita, listen, this stuff is really cool, I'm not going to just leave it at a course, okay? I have extensive background in the world of computers and artificial intelligence and startups and venture capital. I want to meet the people on the biology side. I want to make a new company, a new idea. Who are they? And I said, if I know the name of a company, it's too late. You're at a too late stage. Come to meetups, right? That's where those things happen. Come to networking, come to this. So somewhere secretly in the audience is that guy who's looking for, the yin to his yang of who are the biology people with burning ideas and burning questions, particularly in fields of combining the computers and the biology together, right? Predictive biology, artificial intelligence, neural networks. I don't want to say too many examples because that would limit. It's very, very broad. But if that's interesting, you like talking about it, come to me. I'll make shiduchim. If other things are interesting, can you tell I'm from a religious background, or is it just me? If other things are interesting you, or you have questions, or you wanted this and you're shy, come to me. I know a bunch of people scattered throughout here. I can really make that happen. I think it's much more important than just coming and hearing a speech. You can really do that anywhere. It's recorded, right? That's officially all. Are there questions? Yes.
Basic questions. I'm not a biologist. Let's say you have a genetic disease for another. Let's say someone is autistic. He was born autistic. He has autism. It's part of his genome, and you want to research and understand what part of his genome are causing him to be autistic, and it's more than one gene, probably hundreds or more. How do you do this, and how long does it take?
It's a great question. Did you guys hear that question? Yes. You want to research something like autism, and you know that it's hereditary, so it's something to do somewhere with genes. Which gene is it? How do you go about researching that? To understand which genes are connected to which diseases, researchers need to create models of the disease. You can't poke on humans. So you choose which model is right for you. It can be a zebrafish or a mouse or a fruit fly, each one depending on the needs of your
Why can't you take
research.
DNA from humans?
You can definitely take DNA and change it, but how will you see?
Take DNA and compare it to other person.
Oh, okay, yeah. So in terms of I definitely went on the complicated. Step one would be to compare the DNA of a sick person and of a healthy person, and to see the differences. Problem is, a lot of it's not really at just that letter level. But yeah, basically it's a really, really long thing, and you went with autism, which is one of the hardest ones, right? Because it's multiple genes, and it's behavioral, which means most of our classic models you can't use. Behavior is one of the more difficult things to study, so you have to go with a higher organism, not yeast or single cells, right? And higher organisms take more time to reproduce, take more money, it's complex, and multiple genes in this. So you need a model. It's really hard. Feng Zhang is working on it. I have a friend who made a autistic zebrafish, but I still don't really understand because she's like, they're not social. With CRISPR, okay? So for more details, I'll set you up with her to talk to her. I'm very into that. I really don't know everything at all, but I know generally someone who will. Like you're at most 1 or 2 steps away from your answer with just me. Pretty cool. Yeah.
So are we stuck in the point that we can only engineer new organisms, or we can't fix diseases for an old person, right?
No, that's not true.
No?
But it's much easier for new people, yes. The question is, can you cure diseases in adult organisms? Let's just call them people. Or can it only work for new people, like working on an embryo level at a single cell level? It's much easier to work on new people when they're only one cell. It's pretty much a 0 or 1 situation if you've done it. And when you have 37.2 trillion, way harder, but not impossible. And all of the cures that we were talking about are being developed for adults. One of the indications is cystic fibrosis, which is super important, right? Yeah. Did I not understand the question?
No, you understood the question,
Okay.
but can I have like 2 examples? So except for the cancer, that it was leukemia, so it's through the blood you can reach.
There's a type
all
of
the
blindness
cells.
that they're trying to cure.
How are they doing the blindness thing?
We deliver the gene therapy to the eyes of the adult.
To the eyes directly?
Meaning, okay, don't freak out, but a lot of it is like the HIV virus.
So
but it's okay
potentially-
because we know more.
Potentially, we can target any type of cancer through T cells from the blood, right? Because the blood
They're working on
reaches the
diseases
entire body.
of the liver, of the pancreas.
How about
Absolutely.
diabetes?
And also keep in mind that for some diseases, you don't have to get to every single cell, right? So for cancer, you definitely want every cancer cell to die. But let's say something like diabetes. If I can activate just part of my pancreas to produce insulin, then that's enough. I don't have to reach every single cell. Muscular dystrophy, they're trying to, with CRISPR, fix the protein dystrophin to give people muscle tension. You don't have to get to every single cell. A bit is enough. Same thing with cystic fibrosis. Okay, yeah.
If you really cure only part of the body, not inserting the genes, the correct genes, or fixing the problem in the whole body, can't the person inherit it to his kids?
Tell me that one more time.
Okay, if
The
you
fixed
have
version.
person, yeah, I mean you have a person that has kind of disease, you cured it with gene therapy.
As an adult.
As
I
an adult,
cured an adult.
yeah,
Will their kids still have the disease?
yeah, because you didn't fix all the
There was
problems.
a story of a guy who was suing his wife because his child came out ugly. And his wife is beautiful.
In China.
He had invested $100,000 into that beauty, right? There was like a nose job and a this and all these things. And then the child looked like, her before the surgeries, right? So does that answer, right? Basically, the DNA that we give to our children is not the DNA of just any cell. It's the DNA in our respective egg and sperm cells, right? So if you're not fixing that DNA, and in the case of a man, that would be pretty much impossible because you're constantly producing new ones. If you're not fixing that, then the child will inherit that DNA. Now, it's still okay because there are a lot of diseases that. Why is it still okay?
You can fix in vitro. You can you can apply the CRISPR in vitro.
So now for the person who has
Okay.
that type of mutation and they don't want to pass it on to the children, they can do in vitro fertilization where the egg and the sperm are in a plate and you fix it there. That is definitely possible. Today, that is not yet being done. First of all, we haven't done enough safety tests, whatever it is. There are a lot of ethical issues. What constitutes a disease, right? Maybe I live in a country where blue eyes is a disease. Can I fix that? Where do we end? Do we have augmented children? Again, it's really not something I would worry about too quickly. But in theory, yes. In practice today, not yet. And more common with in vitro fertilization would be if you have a recessive disease to just find the embryo that's anyways healthy. It's much easier than editing but in theory, yes.
The Chinese people did.
They did, but it was so hyped up. It was on embryos that were being thrown away from in vitro fertilization.
Another one after that, right?
There has never been anything that was then implanted and made into a baby, which, let's be honest, that's what we're all interested in. No. Yeah.
Ethical limitations limited progress in the field, and if so, does authoritarian regimes have an edge?
Probably on everything anyway. But do ethical limitations affect us in the field? And do totalitarian regimes have one up on us? First of all, look how much the U.S. owes to China. Like, those guys rock. And it has nothing to do with like ethical limitations. They could just do ethical limitations limits. Okay, this is really interesting. A very short time after CRISPR was discovered, the 2012 time, not the 1980s, they quickly had this emergency meeting summit to discuss the ethics of what is going on. And they said, why did we not do this sooner? The answer was everyone just thought it was sci-fi and that they didn't really have to. And all of a sudden they're like, well, let's lay groundwork, but people were doing these experiments and this and this and this. So I don't know how clear the ethics are just yet, what we're going to be able to do. So far it hasn't really limited us because basic research doesn't do overly unethical things if you're not a vegan. So since we're mostly at the basic research level, it's been okay. And if you're working on diseases, then yeah, I don't think it's actively really affected us too terribly. Do totalitarian places have a one-up? Is there something you really want to do? Talk to me later. Let's go on a trip. Yeah.
What I understand is that the CRISPR comes and cuts the gene, and then in the NHEJ, like it will fix it back itself. So are there any cases where everything goes wrong? Like, I mean, if you're disturbing a gene and it fixes itself, like if it forgets something, there could be some side effects which are bad.
Give me like an example, like side effects for.
I don't know. I'm just thinking off my head.
Yeah.
So like, let's say you're cutting this gene of this animal that you want to remove like glowing properties, and you want it to not glow at all.
Good.
So you cut that gene and then it breaks, and then when it fixes itself back, instead of not glowing, it grows like a third.
It kills everything
or,
it
like,
sees.
kills
Yeah.
everything, or
Okay.
does,
So what happens if I try to, disturb one gene and end up destroying the world? But not really. Meaning, what happens if I expect I expect one phenotype, and I get a different phenotype that's no good at all, right? What's that? I don't think that's what that was. I think they just didn't understand what dinosaurs do, which I don't know why. Meaning, right? Like they just made dinosaurs and then said, "Oh, we don't know how to control these." Okay, so no. But think about this, okay? I want to take this gene away, and CRISPR has done it. Now, what are the chances of something crazy happening as a result? The answer is 0 and also a lot. Why? If there's a bug in one of the apps on my phone, and I delete that app, there's no reason it should be doing anything to the other apps. Like they truly are not related, right? If I've managed to get to one sequence and delete that, there's no reason that should be doing, like all of a sudden popping up with a new phenotype. However, if I deleted this gene, I can't actually use the app example just a second, but if I deleted the gene and actually the protein that it makes interacts with a ton of other things and I've ended up, it's kind of like Jenga, right? So from deleting that gene itself, there really shouldn't be anything too terrible. But if that happened to have been like a cornerstone gene of a crazy pathway, then I might end up, killing the organism, which is why we test things in mice first. Another problem though is, imagine someone who here has an iPhone.
Google campus.
Wow.
There's Android
Okay,
land.
you win.
Android land.
We have this fight all the time. Have I proven that I'm intelligent at this point in the evening? I'm an intelligent person, okay? I have an iPhone and I love
You're
it.
the only one like this.
I'm just saying.
With iPhone.
So
The only
how do you
one
delete an app
intelligent.
on an Android? What's
The
the
only
process?
intelligent one.
Very simple. You delete it.
You go, okay. In an iPhone, everyone here has owned an iPhone at some point in their life. Don't even pretend, okay? You've owned it. So you press down until they all start to shake, and there's that X, okay? Now imagine to yourself if you had like a very fat thumb, and you by accident delete 2. That can have very bad unintended side effects, right? Those apps have nothing to do with each other. The fact that you want to like order food and order a taxi, they're not related. They're just spatially close. You're gonna be in trouble, right? And then you don't even know you deleted it. That's a much larger concern in terms of CRISPR. What I mean by that is off-target effects. So the CRISPR is very, very specific. It cuts where you tell it to, but you have 5 billion letters in your genome. If there's another 20-letter sequence that's similar enough, the CRISPR may cut there too. And functionally, they have nothing to do with each other. The sequences just look similar enough. And that is a big danger, because you're trying to cut one gene, which shouldn't really do very much, assuming you understand its pathway, but you've actually cut a totally unrelated gene, and you did not want to do that. So there are all these tools on the computer that will score the sequence against the whole entire genome to make sure that there's not enough similarity, and that's how you will choose where to target, etc. Obviously, I couldn't fit every single entire thing into this whole lecture, so I would only focus on the amazing things of CRISPR and not the off-targets. But I think that answers yours, yeah. But it's not fair. yeah.
I want to know what's the difference between CRISPR and restriction enzymes.
Oh, that's amazing.
And,
I'm gonna answer you later.
okay.
But in short, restriction enzymes are the immune system that a bacteria was born with, and CRISPR is its adaptive immune system.
Okay. When you tell it where to cut, it just cuts in the middle, or you can tell it to cut the sides?
The code that I give CRISPR, that I give Cas9 to say go there, go cut, that code is a piece of RNA, and it's 20 nucleotides long, 20 letters long. The CRISPR will cut between letters 17 and 18. How's that for specific? It's only going to do that though if that sequence is near a PAM motif. So if you have a lab and you want to do CRISPR, talk to me afterwards, because you don't mess that up. But it's too. But yes, okay. More. Yeah.
We'll give you a number.
Maybe I missed something, but
You came in late.
What?
You came in late. You're not allowed to ask questions. No, I'm joking. Yeah.
I understood in the past that each cell has got all the DNAs, all the copies within. So if you edit, you have to edit all of them if you have some problem?
So that's a great question. Let's say a person has a mutation that they were born with. That mutation will appear in the genome of every cell in their body. If you want to fix that mutation fully, you would have to edit every cell in their body. But practically speaking, you usually don't, because that mutation might affect one part very heavily, their liver, but the same mutation in their eyes doesn't really matter. So you want to only fix the mutation in the places where it's actively affecting. Did that
You
answer?
still has to edit those cells within the liver, yeah?
Yeah.
And it's so simple in terms of
No.
process?
No.
No, after this, of course, CRISPR will be like adult technology or something. Should it be automatically, or somehow it should be done cell by cell? I just don't get it.
So CRISPR itself is a protein and an RNA. The question is, how's it getting inside your cells, right? Is it an injection? Is it a pill? Is it a spray? Is it packaged inside of a virus? Because we know that viruses have a talent for getting past cell membranes. Your question is less about CRISPR and more about
Delivery.
delivery, right? And that really, really, really is one of the top challenges. CRISPR Therapeutics, I think it's them, it might be Editas, has partnered with Novartis, right? So what you'll find is that when you're really, really good at this one technology, but you need another 7 to make an actual product, instead of spending 45 years learning that technology, you're just going to start partnering. So Novartis is great at delivery, and CRISPR Therapeutics is great at CRISPR. So they're teaming up, and they're trying really hard. So I'll keep you updated. Yeah. Very good. The questions disappeared. Because there were more, but I guess you all dashed them. What's that?
No, the European Union is investing in a project to develop a connectome to get to know more about gene function, to get to understand what
The connectome.
the connectome, yeah.
Yeah.
So how is that going to impact all this effort that is being done at this time? I mean, I think that once you can count with that kind of tool, you're going to be targeting your tasks in a much more efficient way, and you can, I think that you will be able to go in steps of magnitude instead of going in steps of order,
Everything. The reason this is all going to work is because everything has to move forward at the same time.
But this is central.
Okay,
Is
our
central.
knowledge about what genes do, our knowledge about interactions, our knowledge about delivery, our knowledge, they all have to kind of reach crescendos at the same time. Yeah, and absolutely, we're all standing on the shoulders of giants, right? These guys can only discover that stuff because someone else discovered PCR, right? So it's all, and the connectome's part of it. It's all going to be part of it. Okay, got it. Thank you so much for all of your amazing patience. And wow, it's really late. And thank you so much also really to Ilya, who honestly organized this thing from A to Z, every single, all of the advertising and the meetup and the event thing and sharing and telling me where to be and when and what to put where and like basically plopping me here. Ilya is one of the founders here of BioHub IL, and I encourage all of you to go talk to Ilya and talk to Karen. She's very tired. Talk to Ilya. We rely on Karen way too much, and I feel bad for her. It's the problem when you do a good job. I've learned that very early on, so I don't do it often. And about BioHub IL and how you can get involved, if you want to learn more about this stuff, if you want to do stuff that's hands-on, if you know people with tons of money who can donate equipment, we need a lot of equipment, or just a lab that's lying around, definitely need that. And